I guess we should wait for maybe a little bit.
No, we usually start exactly on time.
Oh, exactly. Okay.
Yes. So yeah, I started the recording, so just get started.
Okay. Okay. I just
No.
give people the benefit of the doubt here. Anyway, thank you for coming, everyone. So today we'll talk a little bit about RNA sequencing. Today will be mostly about the background and sort of experimental considerations, and then we'll do more hands-on next week. I mean, next time. Oh, and just to clarify, feel free to use the chat. I'll try to check it occasionally, and if you have any questions, you can sort of drop them into chat, and then if I'm going too fast or too slow, or if something is unclear, hopefully we can clarify this. Because this is a really relatively large audience, so it's hard to please everybody without clarification. Okay, so basically, what is RNA sequencing? Here is just a slide showing the basic overview. So you might have your sample of interest, then you extract your RNA, you then fragment, reverse transcribe, and ligate adapters and amplify, so that for each RNA you have an adapter that you will be then putting in a sequencing machine. Then here in this example, we're sequencing paired end reads. So for each DNA or cDNA molecule, we're sequencing from one end and from the other end to get this read pair. And then finally, we get, you have your genome, which is then split into exons and introns. And then you can take your reads and map them to the genome, and hopefully they overlap a particular exon or a particular gene. And then you know that you have X number of reads from gene A and Y number of reads from gene B. And then from then on, what we have is a table of genes and gene counts across different samples. And from then on, you can do all kinds of fun things, such as differential expression, but you can also perform a variety of other analyses. But essentially, if you would summarize RNA-seq, this describes it all. However, you can also look at RNA-seq in a little bit more detail. So there are actually a lot of different steps and a lot of different analyses that you can perform. We're not going to talk about all of them, but some of these I do want to touch upon. So starting with the experimental design, you have to make some decisions, such as what kind of library you're making, the sequencing length, the number of replicates. Further, you can perform some sequencing design. Sort of, for example, some people add spike-ins like ERCC genes. What kind of library prep you're making if you're going to be randomizing across the sequencing runs, and trying to avoid the various confounding in experimental and technical factors. Additionally, you want to perform some quality control. So once you have the raw reads, are those reads of good quality? Read alignment, are the reads aligning properly? Quantification, which is counting the reads. Reproducibility, where if your reads are mapping where your different samples are similar or not similar based on transcriptome-wide expression. With the transcriptome profiling, you have a variety of steps, such as read alignment, where you take, as I mentioned previously, you map the genes to the transcriptome, compare the genes that you're getting to the existing reads, whether the reads fall within the existing annotations. You can perform various quantification levels, whether on the transcript level, gene level, or exon level. There are various quantification measurements like regular counts or PKMs or TPMs. Finally, once you have a matrix, you perform differential expression, which is based on pre-processing, the actual differential expression, looking at expression level of one gene in one group versus a different group. You may also perform alternative splicing analysis, whether one gene has a certain exon or another one doesn't. A common post-differential expression analysis would be something like pathway analysis or functional profiling, where you're looking at if there are functional groups that are overrepresented in your results. You can also perform some visualization, and I think Anna covered that relatively well last week, looking at the different You can look at your genes in the genome browser. You can make various plots there. There's also more advanced analysis that you can make. So for example, looking at small and non-coding RNAs. Well, small RNAs, you probably need a separate library, but for non-coding RNAs, usually you can capture them with the standard library. There are obviously some complications there. You can look at gene fusions that are possible. So if you have two exons from different genes that are now present in the same transcript, there are also long reads and single cell analysis. And then finally, you can integrate it with additional data, such as looking at various variants that might be causing differences in expression, looking at chromatin, such as ATAC-seq, which is really popular, or ChIP-seq for looking at transcription factor binding, and of course, sort of metabolomics or proteomics that you may have as well. So just I think this is a really quick mention that usually when we talk about any kind of sequencing here in this day and age, in this place, is usually Illumina short reads, which here are represented in the black line, where you do the fragmentation, cDNA synthesis, adapter ligation, amplification, size selection, and then finally sequencing. But there also more recently have been more additional technologies such as long-read sequencing with PacBio, where you can actually skip some of these steps and you get longer reads. And then also something like Oxford Nanopore, where you can skip most of the steps and you really just need the adapter, and then you can actually sequence full mRNA molecules as they're floating through these nanopores. And of course, while Illumina short reads are looking at the sort of sequencing by synthesis, so more similar to kind of the classical sequencing methods where each base generates a different color. These new technologies are using current, so you can sort of do, so it's a little bit different OS output. Of course, more recently, there's been modifications to the standard bulk RNA sequencing protocols, which is using single cell approaches. So there are a number of different methods that are available at different scales, you know, performing from sequencing single cells in individual tubes to using plates, to using microfluidics, to using droplets to give you different output. Of course, there are many other considerations for single cell sequencing, but essentially some of the basics are the same, whether you're doing bulk or RNA, where you are just taking particular transcripts in different samples and you are counting them. So just a little bit about the details of the data generation, where you normally take total RNA. The second step might be to remove contaminant DNA. So there are different library types for RNA sequencing, which will impact the results. So for example, you could, if it's poly A selection, you will only select mRNAs just because you're selecting the poly A tails. You might also do total RNA, where you might be eliminating ribosomal RNA based on a ribodepletion kit. You then fragment the RNA to make them to make the individual fragments more compatible with the sequencing instrument, which is Illumina in our case. You then reverse transcribe them into cDNA to make them, again, compatible with the sequencing protocol. At this point, you can actually, depending on how you perform the conversion to cDNA, there are actually strand-specific libraries that are available. So you can, since you are starting with a single strand, it is possible to capture that strand information. So then later when you sequence both strands, depending on which direction the read is going, you can actually recover the original strand. So if you have overlapping genes, for example, you will be able to tell which strand your particular read came from. You then ligate sequencing adapters. Again, for the sequencing, you perform amplification to generate a large enough number of fragments. And then finally, you sequence them on your instrument. There are also some more advanced methods that are possible that are measuring active transcription. And so, for example, you can label the nascent RNAs to separate them from all the total RNA that's present. And then you can really separate, rather than looking at all the RNA, really just the genes that are actively transcribed at any given moment. And there are a number of different protocols available for that. But really, at the beginning, you really should be thinking about designing RNA-seq experiments. And there are three very basic measurements that you can use at the beginning to really define how you're going to set up your experiment. And so here in this review, they're looking at different publicly available data sets, about 1,700 of them from 2009 to 2016, where you can see the distribution of the read lengths. So here they're actually splitting them by single end and paired end projects. And so, for example, in 2009, when the only read length available was the 35 base pairs, most of the experiments were of that size, with the maybe one outlier here at 50 base pairs. And over time, it seems like for the single end experiments, we actually have settled on 50 base pairs as the standard. And then for the paired end, it seems 100 is the most common protocol. Another consideration is the read depth. Again, over time, this as one might expect, there has been an increase in the actual read depth for the experiments, whereas before we had here we're starting at about 1 million, and then it's actually log scale. So although we had experiments as low as 1 million reads, over time we're sort of going to 10 million and maybe even approaching 100 million depending on the experiment. But it's a kind of a wide range. And then finally, the size of the experiment, that's a little bit harder to improve over time. And so again, this is highly variable, whereas obviously there are experiments with only a single sample. I'm not sure what they're doing with that. Not a great idea, but then you can also have, but there are also experiments with hundreds of samples, and all of these decisions will impact what you can do with your data. And just, I'll come back to the long read and direct RNA-seq analysis. So normally what you do is that you have your gene, which is split into exons and introns, and then you have different transcripts that, or isoforms that are present for that gene. You can have, for short read cDNA, or sort of the libraries that we're normally used to around here, you can have reads that are actually ambiguously mapped to one exon or another. You can also have reads that are unambiguously mapped. So for example, here they're only mapped to this particular exon, which is only available in this transcript, or this particular exon, which is only in that, or vice versa for the black ones here. You can also have genes that are ambiguous to the isoform. So for example, here you can have a read that spans these two exons, but you don't know which isoform it comes from. But you can also have reads that are spanning two exons, each of which is only present in one isoform. And so those are unambiguous to a particular transcript. What's possible with long read or direct RNA sequencing is that you can actually capture the entire length of a transcript. So then you really know for each molecule which exact transcript it's coming from, and really kind of giving you another kind of level of analysis. And with something like direct RNA sequencing with nanopore, you can actually get the methylation status, which is obviously provides some additional information that's not possible with traditional short read sequencing. So once you have your reads, what happens then? So this is the RNA-seq analysis workflow, where as usual, you start with your FASTQs or your raw reads. And then there are basically 4 phases that are common to the workflow. In the first phase, you align to your genome and transcriptome with tools like TopHat or STAR or HiSat. It's also possible that maybe you're working with a new transcriptome that you're not where you don't have a reference, so you might use something like something like SOAP de novo, where you're assembling a transcriptome from scratch. It's also possible that maybe you're looking for new transcripts that are not described, and then it's possible to do something like string tie to try to determine based on limited information, sort of if you have any novel transcripts. And then there are also some more modern tools like Callisto or Salmon, which rather than doing traditional alignment to the genome, what they do is really what they call pseudo alignment, which is using various probabilistic approaches. They try to estimate the quantity of particular transcripts based on the individual reads that you're getting. And so they essentially do both the pseudo alignment and the quantification in a single step. You can also, so once you align your genes in a more classical workflow, you can also quantify using something like HT-seq or feature counts. or RSAM. Once you have your counts, you have a certain number of counts for each gene. But every sample sequence differently and has its own properties. So next phase would be to normalize them to make the different samples more comparable to each other. So for example, if you sequence one sample 1 million reads and one sample with 10 million reads, of course you're going to have 10 times more reads for the second sample. So when you normalize them, you essentially equalize the number of the sequencing depth and other properties across different samples. Once you normalize, you also then model or perform differential expression. And so there are a number of different tools that are possible, such as EdgeR or DE-seq or Llama, and then that or CubDiff. And then that gives you differentially expressed genes or transcripts, which you can then use for more downstream analysis. So I know you guys have probably talked about this in previous sessions, but just to reiterate, the FASTQ file, when you get it, it's really just a text file of raw reads where you have a read identifier, a sequence, another line, and then a quality score. So you really just have just a bunch of random reads and you're trying to get some information out of them. And so once you have your reads from a particular sample, it's just a kind of it's basically a hodgepodge of data. And there are a lot of different factors that impact that data, such as technical noise, library preparation, sequencer biases, library complexity. And then what you do is then you take these reads and then you try to align them to a genome or a transcriptome. And then you hopefully get some sort of a quantification in the end. And so during this alignment, many different problems can arise. So for example, you can have multi-mapping reads, such as you have one read that's mapping to two different genes. Now, which gene did it really come from? It's hard to say, or actually impossible to say, but you can make some assumptions. You might want to see is a certain gene expressed or not. So if you have maybe one read that's aligned to that gene, is that gene really expressed? And also there's amplification bias. So for example, you might start with really low quantity of RNA at the beginning, and then you're amplifying it. And then when you're seeing a lot of reads from a particular gene, are those reads because that gene is highly expressed, or are those reads because you just amplified your starting material a lot? And so at the end, you get a counts and an expression matrix for each different sample. And so there are a number of, as you may imagine, there are a lot of different aligners, and some of them you have discussed before in this class that are for different data sets. And so here there's one particular review that sort of looked at a number of different benchmarks. Here is a few different assessments based on different aligners. And then one metric that you can use to compare them is the fraction of reads mapped. And sort of depending on the particular tool you use, the fraction of reads mapped actually varies quite a bit. Some of them are almost perfect, and then a lot of them are much lower than others. So, but this is of course only one metric. And what is the fraction of read mapped? I could technically take any read and say it's mapped well in the genome, but do you really trust me if I'm the aligner? Additional question that you do have is the choice of a gene model or the reference. And so again, this sort of came out before, where you are aligned to the genome, and then within the genome, there's an annotation or transcriptome. And so really, but the different annotations come from a number of different sources. So some of the common ones are, for example, Ensemble or NCBI RepSeq or UCSC. And they all have their own different annotations, and some of them are interchangeable, some of them are not. And so, for example, in this particular study, they looked at the overlap between the genes that are present in different resources, and the differences are quite stark. So, for example, common to all of them are 20,000, and RepGene has very few that are unique to it. But then Ensemble actually has more genes unique to its reference than it has in common with the other two. So which one is the right one? It's a harder question. And then if you look at here, they looked at the different tissues and looking at their mapping percentage to the transcriptome. And then here they break it up into the three different versions. And of course, the number of mapped reads varies quite significantly. So here, for example, this red dot is over 80% for Ensemble, but drops by quite a bit for RepSeq. by about 20 points, 20%. That's quite significant. And then they also use both transcriptome and genome in combination, where once you have the genome information, the two different data sets do become much better, because now that they have the full genome information to supplement just the transcriptome. So what does that mean, different annotations? It's not just about the numbers. Just because one has 2 genes and one has 1 gene, it doesn't mean it's 2 twice better. But there are also different, within specific genes, there are also differences. So here, for example, they compared the ref gene annotation versus the ensemble annotation for PIK3CA, which is a relatively, this is from 2015. It's a little bit old, but it's definitely a sort of in the the modern era of genomics. And PIK3CA is a very well-known gene that many people have studied even before 2015. And then you can see here that, for example, for ref gene versus ensemble, the isoforms are quite different. And if you look at the expression in the liver, it's clearly that the longer the ensemble isoform is present and not the one that's described by ref-seq. So if you were using the ref-seq model, all of these reads that are falling outside of its known isoforms would not be counted, so you're actually losing that information. And another possibility is that you might be only capturing reads in this region, and then in that case, you might have some reads with ensemble, but with ref gene, you might have 0 reads, and so you might think that the gene is not expressed when it is. Of course, in this case, it's UTR, so maybe not as important, but depending on the gene, you can have different combinations of available annotation and across the different versions of the annotation. But even within the different annotation sets, there are different versions. So, for example, GENCODE, which is used as the basis for ensemble now, really has a new release several times a year. So here they're looked at the stats for their releases from 2016 to 2018. So in just 2 years, they had 4 different releases for the human, which are actually just based on the raw numbers. They're relatively similar. But then in their mouse releases, they had 1, 2, 3, 4, 5, 6, 7, 8, 9 releases in that same time frame. And the number of genes has increased quite substantially. And not only did the number of total genes increase, the number of transcripts that are that make up those genes or isoforms increased even more so. So even sort of within this GENCODE annotation, based on which version you use, you are going to get some differences. Although obviously the differences aren't as much as between the different sources of annotation. Another hot topic in RNA-seq analysis that has that has led to many questions is duplicate reads. In order to generate enough cDNA for sequencing, you do need to amplify your sample. But at the same time, PCR does not amplify all sequences equally well. So when you're amplifying millions of different sequences, you might not get the same result. And so the duplicates that in the end, may correspond to biased PCR amplification of a particular fragment, or duplicates maybe are expected. So for example, if your gene is really highly expressed, you should expect to see many very similar transcripts in that gene. But how do you sort of, so you can take 2 different approaches here, but which one is better? And so usually, how do we determine duplicates? Again, I think you guys covered this earlier. So you're essentially looking if your reads or your DNA fragments are the same. And so how do you know if they're the same? They're actually, that's basically if they're mapping to the same exact place in the genome. So for example, here I drew in a situation where you can have single read or paired end reads. So usually for RNA-seq specifically, usually people do perform single read sequencing because it's cheaper and it's usually sufficient. But for example, in these 3 fragments, if you perform single read sequencing, all of these reads would start in the same place. So they all look like they're duplicates of each other. But if you had paired information, you would see that these 3 are actually 3 different fragments. So they're actually not duplicates. So just by going from paired end to single read reads, you are introducing additional duplicates, which are simply artifacts. But in this study, they actually looked at different libraries, and they They looked at paired end duplicates, single read duplicates, but also UMI duplicates, where they added unique molecular identifiers. So rather than trying to determine the duplicates computationally, the UMIs are a part of the library. So then actually when you're making your library at the beginning, each transcript is labeled with a unique barcode. So then you really know that is in after amplification, whether that's a unique transcript or not. And so here, this is a little bit hard to read maybe, but you have on the left the mean expression across different data sets, and then you have dispersion. So here, for example, the blue line is the single read duplicates, and you can see compared to the paired end duplicates in yellow or all the reads in gray, you removing single read duplicates, which includes sort of situations like this, not surprisingly decreases the mean expression. But at the same time, it actually increases the dispersion or the variance of your reads. So, as opposed to using UMI duplicates, which are more real, you can compare the original versus the green, which is duplicate removed, which is a slight reduction in expression as expected because you are removing reads. But then the dispersion is actually reduced much more noticeably. So there you are actually having more consistent signal there. So then it does indicate sort of using UMIs for duplicate removal is better than using sort of kind of more traditional duplicate removal based on where the reads map. So hence the conclusion is that perhaps probably removing duplicates just based on standard libraries is not a good idea. Finally, you get to read counting, which is looking at how many reads does a particular gene have. And you might think it's really easy. Sure, a particular read is overlapping an exon. So then it goes to that exon, which belongs to a particular gene. But you can have all kinds of edge case scenarios. So for example, if a read is over a gene, clearly it belongs to that gene. But you might have a read that overlaps the gene partially, and then partially overlaps an intron. So is that read really from that gene or is it not? It's a little bit arguable. Another situation you might have a read that spans two different exons, but it also spans an intron. Does that read really belong to that particular gene? Because you might say, well, maybe it's a read through or it's intron retention, but that's probably less likely than this read coming from DNA as opposed to RNA. But again, it's a trade-off. Again, you might also have another hand, you might have a read that spans two different exons perfectly. And then that's pretty easy because then it's clearly belonging to that gene. You might also have two genes that are overlapping, but if the read overlaps one gene, it belongs to that gene. On the other hand, what if there are two overlapping genes, but that read overlaps both of them? Which gene does it really belong to? And again, if the overlap is partial, it's probably gene A. But if the overlap is complete, it could be A or B. And so here, for example, if you have strand information, the two genes are probably on different strands, then you can determine from that information whether the read belongs to A or B. And then, of course, you can have a read that sort of doesn't align either, and then sort of where does that read go? And of course, sort of this kind of situation, probably not very common for mammalian genomes like human or mouse, but if you're working with fly, this actually happens quite frequently and has sort of personally caused me a lot of pain in the past. Then you can also perform isoform-aware quantification, which is a slightly different challenge, which is you have a single gene, which consists of different isoforms, and you can actually try to quantify different isoforms and see what the contribution is. So here, for example, we have relative probabilities of different isoforms. And so, for example, here, these reads all fall into this exon, which is present in all isoforms. So you actually can't properly quantify it, or at least at the isoform level, because here all isoforms are contributing to this read. But then on the other hand, you can have some reads, that this one in blue here, where it actually overlaps this exon boundary, which is only present in that isoform. So based on that information, you know that this particular read comes from this isoform. So now this isoform has an extra count, and so now you might be able to deduce that that particular isoform is more highly present in the sample than the green and the red. And so by going through all the different reads and all the different combinations, you may be able to determine these abundances. But what are the broad transcriptomic questions? So usually there are kind of three main questions that you can ask based on the RNA-seq data, which is you have a particular gene in a particular sample, and then you have this gene across different samples, and is that gene detected, or is it meaningfully expressed in your sample relative to some population of samples? More common is this gene differentially expressed? So for example, you have two conditions and you have your expression values in one group versus the other group. And so here, this gene is higher in one which is the other. And then also you can have co-expression, which is are two genes correlated across the different samples. Okay, so I actually have a question, which is about why would you use one reference versus a different reference? Sort of if one covers more transcripts, why not always use that one? And so that's a good question. And usually the answer is yes, why not? You should use the one with more transcripts and more genes. But on the other hand, perhaps there's a reason why there are differences in how genes are annotated, because the reference, sort of the sources that use fewer genes are more stringent in how they define their genes. Really, when we say genes in the context of RNA-seq, sometimes people are confused because sort of not necessarily biological meaning, because gene can mean sort of anything that's transcribed. So for example, link RNAs would be considered a gene, or small RNAs or microRNAs would be considered a gene in this particular context. And so maybe you are interested in only looking at very high confidence genes. And so using kind of a more stringent version of the annotation, you're only limiting yourself to the genes and the transcripts that you're really more sure that they are real and they're not just some kind of noise. And so maybe if you're using less stringent annotation, you are capturing more of that noise, and so that the differences in the end that you're seeing are actually just the same noise that the authors of the annotation found. And speaking of noise, sort of why do you need more reads? And so really, the sort of the concept that becomes apparent that sort of been used is sort of the Poisson distribution, which is here you can look at the twofold differences between the mean read counts between two different samples. And so for example, here we're looking at one read versus two reads, where sort of if you're randomly sampling these reads, the chance that the two distributions are overlapping are quite high. You most likely will capture one read in the blue, and then most likely two reads in the red, but it's going to be variable. So once you move to 10 reads versus 20 reads, so you're sampling, instead of sampling once, you're sampling 10 times, all of a sudden the distribution becomes a lot more clean, or again, you're still likely to get 10 reads in this one and 20 reads in the red, but you might actually get 15 reads in both sometimes. And then while you have twofold difference, just by chance, you're actually not fully capturing, you might not actually capture that difference. And then finally here, if you go to 100 reads, or sampling instead of once or 10 times, you're now sampling 100 times, all of a sudden the distribution becomes a lot more narrow. So here at 100 reads, you have a very clear peak in the blue sample, and then at 200 reads, you have a very clear peak in the red sample. And so there's, if you look really closely, there's a very minor chance that this sample actually has closer to 200 reads, and this sample might have closer to 100 reads. But the overwhelming majority of the time, now you see a very clear separation between the two distributions. And so basically the variance is higher relative to the total count when the counts are low. So by increasing the number of counts, you're essentially increasing the number of times you're sampling your reads. And so the more times you sample them, the more likely you are to capture the real level of expression. And so there are actually a number of different differential gene expression tools that have, and statistical tests that have been developed over the years to sort of try to address kind of what's the right way to compare different groups and a different number of reads across different groups. So you can use sort of the classic t-test, which assumes a normal distribution, which is maybe not accurate for RNA-seq, which is should be negative binomial, but that is one kind of a classic test that you can use, and then you can just see if the two samples diverge from each other. They're also you can adapt it for the log normal. You can use Mann-Whitney, and then there are kind of a number of more specialized tools that exist that sort of try to modify the known statistical tests to fit the distribution that's expected from the RNA-seq data. And so you can actually look at the various statistical properties. And here in this particular paper, they used EDGE-R as the test, but the different tests are relatively, we can assume for this that they're relatively similar. And then using the statistical properties as a function of the fold change cutoff and the number of replicates of the NR. And so, for example, in this first plot, you can see sort of the fraction of significant genes as you increase the number of replicates from 2 to 40, which is quite high. You can see that the fraction of significant genes really stabilizes. Looking at the different significance cutoffs, the fold change cutoffs. So when there's no cutoff here in the blue is 0, the false positive rate, true positive rate is slowly increases over time, but then if you actually just add the fold change cutoff right off the bat, you actually do have better results because if there's a really high difference, then it's more likely to be real. And then here there's essentially trying to combine all the different metrics. So essentially, so while the true negatives stays relatively constant, depending on regardless of the number of replicates, as you add the replicates, you're actually increasing. Over time, you're increasing the true positive rate, which approaches an asymptote here, and then the false negative rate, which substantially decreases over time. So if you want better results, you should definitely add more replicates to your experiment. And there are also different normalization methods in terms of once you have your raw counts, what kind of as I alluded to previously, you may not necessarily you probably shouldn't look at them directly. And so there's a very simple normalization, which is just total counts, where you can take your gene counts and you divide them by the total number of mapped reads or the library size, and then multiplied by the mean total count across all the samples of the data set. So essentially, you're normalizing them to the total reads, so that one sample that has 1 million reads is going to have, after normalization, the same number as the one that had 10 million reads or 100 million reads at the beginning. You can also slightly more advanced would be the upper quartile normalization, where the total counts replaced by the upper quartile of counts. So you're essentially eliminating the lowly expressed genes, which might be more noisier. A variation of the total counts would be the median counts, sort of instead of looking at the total, where you might have one highly expressed gene that's overrepresented and really drives up your library size. Looking at the median is you're normalizing to a kind of a more sort of a more average value. Also, there are more advanced normalizations. So, for example, DSeq and TMM. TMM is the one that's implemented in the EdgeR package, which use kind of a more complex method where they're really they're using a scaling factor that's for each gene, it's the read count over the geometric mean across all lanes or all samples. And so they're assuming that most genes are not differentially expressed, which may or may not be the case for every experiment. And then sort of the median of this ratio for this lane or sample provides an estimate of the correction factor that should be applied to all the read counts. And then the trimmed mean of M values, or TMM, in EdgeR is somewhat similar approach. And then one that you guys you may have heard about that's relatively common is RPKM or FPKM, which was very popular at one point, which was initially introduced to facilitate comparisons between genes within a sample. And so it actually combines between and within sample normalization. So it actually normalizes reads per kilobase. So it rescales to the gene counts, but it also corrects for differences in both library size and gene length. So per million reads, meaning the per million reads in that particular library. So of course, because different genes have different biases, people have moved away from this particular normalization. And I think Tasha's doing differential expression next week. So I think maybe you guys will discuss the different normalization options in more detail then. And there's also here's a summary of different comparison, summary of the comparison for different normalization methods, where plus means satisfactory. And so depending on your application, so looking at sort of the false positive rate is probably the most important aspect, which is does it accurately represent the difference expression between different groups. Here, DSeq and TMM, which are specifically designed for normalizing RNA-seq data, not surprisingly perform very well, and other methods which are more simple perform less well. But at the same time, if you just want to look at the distribution, all of them are relatively similar enough. And that was actually all I had for today. I thought there might be more questions or comments, which you may have now. And then next week we'll do a more slightly more hands-on tutorial to try to get some RNA-seq results based on some data that you can all do at the same time with me. And so if you have any questions, please let me know, either now or after at some point. Thank you. Okay, so yes, we have a question, which is how to correct for batch effects in normalization. So batch effects is a topic that does come up occasionally. So there are multiple ways of dealing with batch effects, and maybe you will deal with it. Will you talk about it a little bit next week? For in terms of differential expression. So for example, if you are using a standard differential expression tool, a tool such as DSeq or EdgeR, you can actually, in your model formula that you're using for your comparison, when you're looking at two different groups, you can actually include a covariate. So if you have a batch, if you have a known batch covariate, you can include that. So you might want to do that, for example, if you're looking at patient samples, where there's going to be very big variability, even if you do everything perfectly between different patients. But if you have, for each patient, you might have untreated or pre and post treatment, and then you can use the patient as the covariate. And so essentially you're looking at sort of taking that into account. So then a gene might be up in all the patients, but because all the patients have different baseline levels, you would not capture that without kind of adjusting for batch effects. They're also, so that's really the ideal method because that's sort of taking into account doing a statistical test. But then there are also specific packages that exist, such as Combat, COMBAT, or SVA, which are tools which use, which you can give them, which you can input your expression matrix, and then you can specify the batch, and then they will then adjust your expression matrix to, it'll alter your data. Yeah, as Stash pointed out, it's best not to is to avoid batches in the first place, which is yeah, it's, if you can do that, that'd be great. But yeah, because again, if you're adjusting for a batch, yes, you can try to fix it, but you really can never be sure whether the differences you're seeing are a true batch effect or is a biological effect. And maybe if you have hundreds of samples, you might be, that's one case, but if you have very few samples, sort of the normal biological variance is really going to play a huge role in that calculation. Yeah, okay, so then another question is the DE-seq vignette calls for unnormalized or unestimated count data, which is correct. So that this means if one is using that, should they skip some of the steps and just put the raw count in? Yes, so DE-seq does take the raw counts. So once you have the raw counts, you can put that in, and then it will do the normalization, differential expression, and visualization based on that. So if you have output of Callisto or something like that, that's a little bit different because Callisto doesn't actually give you the raw counts at the end, it gives you estimated counts. So they're not, so it's not identical. And so there's actually a package called TX import, I guess transcription import, which will take output from a number of different tools, such as Callisto or Salmon, but a few different others. I don't remember all of them. And it'll actually summarize that data and put it in the format that's acceptable for DE-seq. And actually, just another item I forgot to mention is that Callisto or Salmon by default do output transcript counts, not gene counts. So TX import will also convert transcript level estimates into gene level estimates. And if you want to perform differential expression on transcripts as opposed to genes, that's actually a slightly, a very different workflow than the gene one. Yeah, and then as somebody pointed out, Sleuth is another R package specifically for Callisto. And will the slides be posted online? I think the slides are posted online. I'm definitely happy to do that. I think David Fenio is in charge of the website. So I just can't guarantee when exactly that will happen.